Major histocompatibility complex molecules, or MHC. There are two types, MHC class 1 and MHC class 2. MHC class 1 is characterized by having one peptide leg bound to the phospholipid membrane, whereas MHC class 2 has two legs. MHC class 1 is found in all nucleated cells. Now this of course means they are not found in erythrocytes, the red blood cells. And MHC class 2 is found basically only on antigen presenting cells, and we will see shortly why. Now MHC class 1 can be found in all nucleated cells such as leukocytes, the lymphocytes, such as the B cells or the T cells. They can also be found on phagocytes such as the macrophage, and even on the nerve cells, the dendrites. MHC class 2 found on antigen presenting cells such as the phagocytes, the macrophage, and the dendritic cell. So basically, when an antigen of a pathogen is presented on the MHC class 1 molecule, the outcome for that particular cell is death, because this cell is usually infected. However, when an MHC class 2 molecule presents its antigen, it will activate the T cells, and therefore the cell-mediated immunity of the adaptive immune system. Now let's take a closer look at the MHC structures. So here are some cells, and this cell can be any nucleated cell, even an antigen-presenting cell, APC. And this cell on the opposite side is a cytotoxic T cell, a cytolytic T cell, or a T killer cell, which kills infected cells. Now the structure of the MHC molecule is quite complex. The MHC class 1 molecule consists of 4 microglobulins, one of which is denoted in red, which never changes its structure. Now this is the major histocompatibility complex class 1 molecule. And the top portion consists of the alpha 1 and alpha 2 regions, which acts as a cleft, the binding site for a particular antigen. And the bottom half, alpha 3, and the red beta 2, the domain that stays constant. So this cell will only present major histocompatibility complex class 1 if it is infected, and if this cell needs to die. So here we have an antigen that fits the specific MHC alpha 1, 2, and 3 complexes. The T killer cell has a special receptor, also consisting of alpha and beta domains, where the alpha 3 and the alpha of this receptor are able to bind. Now this receptor is known as CD8 receptor, which is special for this particular type of T killer cell. What it does is that it checks if the MHC being presented is actually an MHC class 1. And, of course, if it is, and everything is good, this infected cell will die. And the cell-mediated immunity will do the rest, which are essentially the T cells, the T killer cells. The major histocompatibility complex class 2 molecule, on the other hand, are found only on antigen-presenting cells, or APC, and presents its antigen to the T helper cells, like this one. The MHC class 2 molecule differs from that of the MHC class 1. It has 2 legs attached to the membrane, but it also consists of 4 microglobulins. One side, however, consisting of alpha 1 and 2, and the other beta 1 and 2. Whereas you can see the alpha 1 and beta 1 domains are the binding site for a particular antigen. Now all the alphas and betas are special to fit only a particular antigen, and therefore this MHC class 2 molecule is special only for a particular antigen. And you might have realized that I used alpha 1 and 2 on both MHC class 1 and MHC class 2, but they are all completely different in structure. It only carries the same name because it's from the same group of proteins. So this cell is not presenting MHC class 2 because it wants to die or because it's infected, but because it wants help from the T helper cells. And once the antigen is presented, like this one, it will bind, the MHC class 2 will bind to a special T helper receptor called the CD4, this one here. And this receptor has 4 parts, the D1, D2, D3, D4, with the D1 and 2 bind to the beta portion of the MHC class 2 molecule. And same like the CD8 receptor, the CD4 receptor checks if the MHC molecule being presented is an MHC class 2 molecule. And essentially, if everything is okay again, the T helper cell is activated and will initiate the cell-mediated immunity, where the T cells begin proliferating and activating the B cells. So just like before, with the MHC class 1, this is a cytotoxic T cell, cytolytic T cell, or the T killer cell. And this is any nucleated cell which is infected by a virus or pathogen or whatnot. Let's say it's an infected macrophage. Now the MHC class 1, as mentioned, consists of alpha 1, 2, and 3 domains, as well as the unchanging beta 2, which represents it as the MHC class 1 molecule. The alpha portion is always different, and this receptor is special only for a particular antigen. Once an antigen is presented, such as this one, the T killer's receptor, called the CD8 receptor, binds to the alpha 3 and checks if this MHC being presented is an MHC class 1. And if that's okay, another receptor called the T receptor, consisting again of 4 microglobulins, alpha and beta domains, checks the antigen. Well, it doesn't check, but same as the MHC molecule, this T cell receptor fits only to a particular antigen, and therefore the alpha and beta domains are always different for every T killer cell. So again, the CD8 receptor checks the MHC molecule, and the T cell receptor checks the antigen. And of course, if that's all good, and luckily this is good, this cell which is infected or which presents the MHC class 1 will die. Now the antigen presenting cell presents its antigen to the T helper cell and uses the MHC class 2, made up of one side alpha 1 and 2 and the other side beta 1 and 2, which is always different to each MHC class 2 because it only binds to a particular, because each bind to a particular antigen like this one. Now the T helper cell's CD4 receptor then checks if this MHC molecule being presented is an MHC class 2. And let's just say this antigen presenting cell is a macrophage, which has digested and already presented its antigen. Once the CD4 receptor checks the MHC class 2, it then follows with its T receptor to check and bind the antigen, to bind to the antigen and check if it fits that particular antigen. Now the T cell receptor structure is same to that of the T killer cell T cell receptor structure, consisting of beta and alpha domains. Fun fact. So next, and if that's all good, it will initiate the adaptive immune response, humoral, and the cell-mediated. So next, let's see how the MHC class 1 and MHC class 2 molecules, how they get presented on the cell membrane, and how they get synthesized briefly. Thank you.